The Wizard of Menlo Park. It's about Thomas Edison that electrocuted an elephant in the early 1900s. An elephant named Topsy. They attacked 3 different circus trainers, and they decided they were going to kill this elephant. So initially they were going to hang it, but they couldn't build scaffolding big enough, I guess. And they decided they were going to zap it. And back in that time, the War of the Currents was going on between Edison and Westinghouse. And Tesla was an understudy for Edison for a while, and then he went to work for Westinghouse, and that kind of fueled the fire between Tesla, right? Yeah, it's brilliant. So Edison was using AC current, Westinghouse's current, to zap stray animals and electrocute things just to prove his current was safer than Westinghouse's. And that's what this one's about. Happy little song about electrocution.
Welcome back, folks. Today we have a very special guest from the Wolfson Centre for Mathematical Biology at the University of Oxford, Professor Philip Maini. Thank you very much for coming today, Philip.
Thank you.
So your group itself, I mean, you don't focus on a particular area of biology in terms of your research. Can you just tell us a little bit about the different things that you are interested in?
So generally what we focus on is looking at the cell level and then look at how cells respond to signals and generate signals. And then what does that mean at the tissue level? And so then you can break that out a number of ways. So you can consider the cell as a black box and say that if I know that a cell reacts in this way to a chemical or in this way to some physical cue, what will the collection of cells do? And then you can also break it down and say, well, let me look inside the cell and try and understand why it behaves the way it behaves. Then, of course, what you really want to do at the end of it is maybe try and link all that together and have a multi-scale model.
So can you give us a specific example of a model that you work on and just explain to us?
Yeah. So one example I can give is work that we're recently doing, which is neural crest cell invasion. So these are cells that very early in development leave the neural crest, and then they have to go to, depending where they leave the neural crest, they have to go to different parts of the body. And we work on cranial neural crests.
So just to explain to people, neural crest cells generally go into the body and then turn into nerve cells.
Yeah, that's right, yeah. So why this is very interesting is these cells move long distances, and they're also the precursors of many aggressive cancers. For some reason, in early development, they're moving and they're doing their thing correctly, and then if they become cancers, then obviously they're moving long distances but not responding to things correctly. And so one of the issues there is, well, what is causing these cells to move? One theory is that the overlying domain produces a signal called VEGF, and that as the cells emerge, they consume VEGF. VEGF is a chemoattractant, which means that cells move up a chemical gradient. And so, as a verbal model, that actually sounds perfectly sensible. Mm-hmm. So you have a self-induced gradient, causes everything to move. But if you try and write this down mathematically and put in the actual numbers, what you find is that by the time a certain number of cells have invaded the tissue, they've used up all the VEGF. So the new cells coming out have no signal, so it doesn't work. So then we asked the question, what would those newly emerging cells see? And all they would see is their friends in front of them. So we said the hypothesis, well, let's suppose they grab hold of the friends in front of them. So they respond to their friends, not to the signal. Colleagues who work with at the Stowers Research Centre looked at gene expression. What you find is that it works. So there's a definite model prediction that the front cells and the back cells are different. So then we got together with them and we designed experiments with them. as a way to test, then the model can make predictions of what would happen if you took tissue from one part of this trail of movement and put them into a different part of a host. What we found was some of the experiments agreed precisely with the model. Some of them didn't agree with the model, and in fact, I usually find that when things don't agree with the model, that's when you really learn. So one of the things we learned about this was that the cells actually change their phenotype with one of the experiments, because the model predicted that if the cells are fixed as leaders or followers, they'd behave in a certain way, and that wasn't the way they behaved when they did the experiment. And the way you could explain the way they behaved in the experiment is that the cells get their cue from the environment. I mean, this is something that the IMO have been, I think, the leaders, people like Bob Gatenby, in saying that you've got to look at the whole environment, not just look at the cancer and say, well, right, here's the cells, the bad things, let's just get rid of them. That it's an ecosystem, and that you've got to look at how the cells interplaying with the environment, and you can see how and it leads to a very simple view of, like, the anti-angiogenesis, the idea that when tumour cells begin to grow, they need a vasculature, they create a vasculature, there's something called angiogenesis, and the idea was that if you reduce the number of blood vessels, you'll kill off the cancer. That would be fantastic if that's the way it had worked. But I think what people hadn't realised in those days was how complicated and subtle cancer is. And I think actually this is one way where a mathematical approach can really help you, because mathematicians are taught to think in extremes. And so if you were to say, think about the idea, if I get more and more vasculature, then the tumour is going to grow more and more, because it's going to get more and more nutrient. So I should reduce the vasculature, and therefore the tumour will get less nutrient and will grow less. Now, a mathematician would not accept that, because a mathematician always thinks in weird ways, and a mathematician would think the following way. She would think, suppose this vasculature is such that a lot of it is concentrated in one place and there's not much of it elsewhere. Then all the nutrient would be concentrated in one place, and only those cells could grow. So the majority of cells couldn't grow. And now suppose you went in and you did anti-angiogenesis therapy and killed some of those vasculature. You'd release the oxygen to the larger community of cells, and you'd actually increase the growth. So a mathematician would say, no, it is not right to say decreasing vascular growth will decrease tumour growth. And what do we find now? We find that decreasing vascular growth can lead to increased tumour growth. And we can use that by now applying chemotherapy. And you have the double whammy of you've got better perfusion, better flow of the chemotherapy into the tumour, and those cells are dividing because they're getting lots of oxygen. Then you kill them. So what you've learned from that is that it's not a simple disease. There are lots of aspects to it, and that's why I would argue that you need a mathematical model, because mathematics is a language, and so we're chatting to each other in a language at the moment called English, and English and our verbal reasoning and our intuition is linear, and that's why we come up with these ideas about reducing angiogenesis will reduce the vasculature, because we think in a linear way. Mathematics starts to extend your intuition into thinking about subtleties, and when you've got a system that's as non-linear and as complicated as biology, we only have one language that can deal with that, and that's mathematics.
There are some trains of thought now that big data is going to be the answer to everything. So why bother focusing on these little mechanisms that you in particular are studying?
What I would say is that there shouldn't be the big sort of hysteria at the moment about big data. The hysteria should be when we start getting good data. That's what's important. And what I'm worried will happen is that people will try and understand these big data, and some of it will be experimental artifact, some of it will be noise in the system that we don't know how to deal with, and at the end of it all we will still not understand the mechanism. The mechanistic models that I'm talking about, to be validated, require you to be able to focus on how things change over time. And traditionally that's been very difficult to do in a biological context, because basically you have to kill the system in order to obtain data from it. Now with advances in imaging and their staining techniques, I think we're in a very exciting part of science, because we're now in a position where it will be feasible over the next few years to validate these mechanistic models. And then once you understand mechanism, that's much more powerful than understanding a correlation. We know there's a correlation between smoking and cancer, lung cancer. So you lower smoking and then you lowers lung cancer. But suppose we could find out the mechanism of what causes this correlation, then people could continue smoking as much as they want, as long as they then took the tablet or whatever it was that reversed the effect of smoking. And I must say this is a fundamental difference actually between biology and mathematicians. The word understanding means different things in biology to mathematics, because in biology understanding means description. So I understand limb development when I can describe the different Hox genes that are switched on in different parts of the limb during development. Mathematician would say that's posing the question, and the work starts from there. And then this is why the statistical approach and the big data approach are so enticing, because they don't explain anything, they describe stuff.
Another thing I was just thinking is that this would probably be interesting to people who are obviously trying to argue against having animals in science. And one of the things that a lot of the anti-vivisection groups will say is that, well, you guys are now making computer programs, you can model what's happening within an animal or a human being without having to use them. How close are we to actually being able to do anything like that?
Well, I think the key thing is model validation. And I think that to date there are very, very few mechanistic models that have been validated. There are some models of ion channels in the heart that in Oxford have been validated to the extent that some of the drug companies have incorporated the modelling as part of their in part of their sort of drug assessment. So it's beginning to happen. But the real issue is the validation of the models. And model validation is a whole area of mathematics and of statistics in itself. I don't think mathematical modelling will, in the near future, replace experiments, but what it can tell you is how to do more informative experiments, and therefore reduce the number of experiments that are done. But I don't think you, at this stage, can get away from continuing to do experiments. But it still leaves the issue that then you are still trying to understand what happens in an animal that isn't a human. So one thing is actually, my interest has changed, because when I'm trained as a mathematician, and what I found was that in developmental biology, which I actually think is a beautiful subject, because scientifically I think is a very beautiful subject, it's a very challenging subject. And I was very interested in the mathematical side of the sort of equations and all that came up. But now I'm finding my interest is changing and becoming more into things like model validation and trying to really link with experimentalists on the work that they're doing, and really trying to get new insights into the biology using mathematics. So my interests have changed over the time, and I think that then one just realises how complicated biology really is, and that one has to sort of draw back one's ambitions about what's achievable in a certain length of time. I'll tell you one thing that's not actually to do with science, but is to do with the way science is done. So when I came back to Oxford, I was about in 1990, And I was sitting at lunch one day, and then some of the older colleagues were talking to each other, and they were paraphrasing Planck's statement about the advance of science, and the paraphrase being that science advances one funeral at a time. It's a longer statement that he made, which was along the lines of, when you come up with new ideas, you can't convince your colleagues about these new ideas, and you have to wait until they basically leave the scene before new ideas can come on the board. And I can remember listening to this, eavesdropping on this, and thinking to myself at the time, that is the most stupidest thing I've ever heard. 23 years later, I think it's the most honest and the most perceptive thing I've heard. The fact that, why can't somebody not be big enough to say, I was wrong, I thought it was happening this way, now I think it's happening that way. And having asked that question, I know the answer to it. The answer is they would never get funding again.
A great kind of insight into how science works, certainly as far as the qualification goes, is a comic called Piled Higher and Deeper.
Oh yes, that's right.
So basically the acronym is PhD.
Yes.
And it's amazing. Anybody who's got a PhD, you'll ask them, they'll say,
right.
Yes, it hits the nail on the head every single time.
That's right, yeah.
It's brilliantly funny, and it's a great
it's
insight
so
into these kinds of topics. Yeah.
That's right.
Thank you very much for speaking to us this evening, and we hope you have a safe and productive stay in Tampa, and have a safe trip back to Oxford.
Thank you very much.
We'll see you next time.
Colourful background?
Nope. I was rescued by the army once.
Really?
When I was 10 or 11 years old in Belfast, so me and my friend, we went off, and then they said, parents said, right, go up this, we want to go up this mountain. They said, right, come back by 5 o'clock. So we wandered off, and then, of course, it's the sort of thing that you think, well, we just get to the top, and then when you get to the top, there's another top. And then this big army truck came up. This was during the Troubles, like 1970s. And then they stopped next to us, and the guy got out and said, "Is one of you called Philip?" And that was when we realised we were in trouble. We got stuck and put into this army truck. And what had happened was the army was passing by, and my dad just waved them down.
Those African grace is in already made. Long days of the homeland days. The Canada reads, they live and bear. Stand where the beast still fails. Bones on parade, what a mess he made. No one is safe from what you created, and the wizard that apart sleeps tonight. Death on his eyes, and the beast whose guilt is least is resting blind to his own crime. Oh, yeah.
What if our universe is sitting in a lab at a university somewhere that got defunded? And it's like all grown over with vines, and everybody's forgotten that it's going on.
And the wizard that apart sleeps tonight.
You've just been listening to a Two Scientists podcast. Now, if you'd like to keep up with our new releases, you can follow us on Twitter at 2SCIS, Facebook or Google Plus using the handle 2Scientists, or for the more old school among you, you can check out our website at 2scientists.org. Thanks for tuning in. I haven't seen anything compete with
Quite
that
impressive.
yet.